Day for Animals in Laboratories. I hope you can hear me okay. My name's Rachel. I'm the Campaigns and Communications Manager here at HRA, and we really appreciate you joining us this evening and for your time and interest in the subject. I know that everyone might be a bit webinared and zoomed out at the moment at this point in the lockdown, but I really think that you'll find the webinar valuable and learn something new. I just wanted to give you an overview of how this evening's going to work. So the webinar will be 1 hour long, and we're going to end up with a Q&A with Andre, myself and Adam. Some people have provided their questions in advance, but if you have a question that is not being covered during the course of the webinar, you can ask in the chat function at that point at the end of the webinar. You'll notice that we haven't got audio or microphones for participants, and this is really due to some security risks because there have been some incidents of offensive online gate crashes with Zoom, so we just want to avoid that for everyone's benefit. As Andre said, the webinar will be recorded, so if you have technical problems or the number of participants exceeds 100 people, which I don't think it will do, we do have a backup because we have this recorded, and we really hope that you'll share it with friends and family afterwards. And if there is an interruption in the transmission, please stay online and wait a few seconds for the link to be re-established. Next slide, please, Andre. So who are HRA? So as most of you are our supporters and members, you will be aware of our mission, but for anyone new, welcome. And as you can read here, HRA is a not-for-profit organisation that challenges the use of animal experiments and promotes more humane and scientifically valid non-animal methods of research. And we would really like to thank all our supporters whose support enables us to operate. We've been kept very busy during the shutdown. Obviously, there are some restrictions as to what we can do in terms of outreach, events and lobbying, but we are using the time to work on new case studies. And we've been making inquiries about animal research protocols in Australia during the lockdown and promoting non-animal research funding opportunities to the relevant bodies here. So we're making sure that we use the time as productively as we can. Next slide, please. And we have 3 panelists here today. Our main panelist is Andre Menache. He's the CEO of Antidote Europe, a zoologist and a veterinary surgeon. He has a particular interest in medical law and was instrumental in amending the Declaration of Helsinki. Dr. Menache's scientific opposition to animal experimentation led him to uncover serious methodological shortcomings in human experimentation, about which he has published several papers. Andre is an advisor to HRA and many other organisations, and we would like to thank him for his generosity in sacrificing his time to prepare for and present this webinar, as well as his ongoing assistance to HRA. And I'm Rachel. I've introduced myself previously. I've been at HRA about 6 months. I've been following COVID-19 research quite closely, so I hope I can assist you with any questions that you may have later on. And I will let Adam introduce himself.
Hello, my name's Adam. I am in charge of operations and administration at Humane Research Australia. I've been here for about 6 months as well, and I have a background in administration.
I've just noticed on the chat, Andre, that the slides are not visible to the participants.
Have we lost Andre? I can't see him connected anymore.
Yeah, Andre may have potentially dropped out, so we'll wait for him to come back and he'll share his slides when he returns. Hopefully it won't be too long. Sorry about this, everyone. Hopefully Andre will be back before too long. Sorry, did you want to continue with your introduction, Adam? I interrupted you. I think Andre's back.
Okay, I'm very sorry about that. We seem to have had a technical problem. So I think what we'll do is we'll begin again, if that's okay with you. Can you all hear me?
Yes. Yep.
Yes.
Okay. Okay.
So all the participants just sign in again with the same link.
Have they? Well, it looks like this. We have 26, so I'm not sure that they've been cut off.
Okay. Yep.
So
Maybe
they
if
may
you
still be
just,
able to hear us.
yep, just waiting for your screen sharing. Yep, there we are.
Okay.
Perfect.
Okay, we were on the third slide, I think.
Oh, so you guys managed to move ahead?
Yes. Yep.
Okie
Yeah.
doke. Okay, so right. Let's see.
Thanks, Penny. So I've introduced yourself, Andre, and myself, and Adam's just introducing himself now.
Okay.
Yeah, hi. So my name's Adam. I am in charge of operations and administration at Humane Research Australia. I've been here for about 6 months, and I came from a background in administration, previously in performing arts in the theatre. That's about it. Yeah.
Next slide, please. So this is the latest statistics, which you may have seen from 2017, which are the most recent that we have collated, and they are a conservative estimate. You can see the state breakdowns there. The total number of animals used in all research in Australia in 2017 was just over 20 million, but a large proportion of that was observational research, which is not invasive. So in terms of invasive research, that was approximately 3 million animal use, which was an increase on the previous year. Next slide, please.
Okay, so I'm Andre Menache, veterinary surgeon and scientific advisor to HRA. Since we are in the middle of a coronavirus pandemic, we thought it would be appropriate to relate this to World Day for Animals in Laboratories. So there you have what a coronavirus looks like. For those of you with a background in biology, it's a single-stranded RNA virus. Of course, it's a lot smaller than that, but that's roughly to give you an idea of what it looks like. So really, it's human activity that has got us into this mess in the first place. A lot of it has to do with human beings encroaching on natural habitats. It has to do with human beings crossing some of nature's red lines and getting too close to wild animals. And then we make ourselves susceptible to what are known as zoonoses, which are diseases that we can pick up from animals and that we can also transmit to them, but it's usually animals to humans. This can include parasites, viruses, bacteria. So this is a sort of a resume of the factors that give rise to some of the viral diseases that we pick up from animals. So keeping wild animals in captivity, which is highly unnatural, close contact between humans and wild animals, overcrowded or unsanitary conditions, the destruction of natural habitats, and then the illegal and the legal wildlife trade. Those are just some of the issues. So as an example, if we look at how we've domesticated ducks, in some cases we've taken them out of their natural environment, which is in the water, and we've forced them to live on land, and we've put them in cages. Now ducks and other bird species are natural carriers of human flu virus. And as long as we respect each other's red lines, then there's no problem. But once humans take these birds out of the water and cage them, then the virus that naturally would pass from one bird to another in the water harmlessly, because these viruses don't harm these birds, these viruses find themselves now unable to do that. They can no longer be transferred through water, so they mutate. They mutate so that they can be transmitted, for example, by aerosol, by droplets, or through the air rather than through the water. So by domesticating ducks, we've now taken a virus that was harmless, living in birds, and we've forced the virus to mutate so that it can be transmissible via air. The next red line that we've crossed is mixing different species which in nature do not necessarily come into contact with each other. So in the slide, you can see there's a dog on the right-hand side. You can see some chickens, you can see a pig. And so what this has created is an environment in which viruses which normally would never come into contact with each other now can come into contact with each other, and again take little bits from each other, and also this is an environment in which viruses can mutate. And then the issue of factory farming, in other words, crowding lots of animals together. So needless to say, this is highly unnatural, very unhealthy. And so we are now at the point where we have a virus that is transmitted by the air, it's mutating, and normally in nature, it's in the interest of the virus not to kill its host. So what viruses do is they can make you sick, but they don't want to kill you, because if they kill you in nature, especially in natural conditions where animals are spread out over the land, then that virus will die out. And in fact, it's committing suicide by killing its host. So viruses normally, it's not in their interest. Natural selection has taught viruses not to kill their hosts. But when a virus finds itself in this sort of situation, where it can pass from one animal to the next very, very quickly, then it can afford to become very pathogenic, very virulent, and far more dangerous than in nature. And so now you can see we're sort of coming full circle. We've got a virus that's air transmitted, it's highly pathogenic, and it's picked up bits and pieces. It's got a bit of pig virus in it, a bit of chicken virus in it. And now because it's in contact with humans, well, then it just needs that last step to mutate to be able to infect humans. And that is probably, I say probably, we don't know for sure yet, but that's probably what's happened with the coronavirus. So of course, wet markets are another ideal source of infection, where again, we are crossing nature's red lines. We are coming into contact with wild animals, consuming wild caught animals, and this has led to problems in the past. People, as the saying goes, history doesn't repeat itself, man repeats his mistakes. So we still haven't learned from that. It's unfortunate to hear from countries like China that the ban on consumption of wild animals and wet markets may only be temporary. So we still haven't learned from our mistakes. And then, of course, the destruction of wild habitats, that does two things. First of all, it concentrates animals, it makes them, it reduces their environment, their living habitat, so they are forced to come closer and closer to each other, which is unnatural, and which means it's easier for viruses to pass from one animal to another. But also, it sometimes forces them to go towards villages and towns looking for food, because we've destroyed their habitats. And then that again sets up a situation where animals and humans can come uncomfortably close together, which they shouldn't under natural circumstances. And then legal and illegal wildlife trade, for example, reptiles and snakes, who normally all carry salmonella and other bacteria and viruses. And again, the last thing we should be doing is keeping snakes as pets. Most pets, like snakes, are not dogs and cats. Many people do not have a clue how to look after them. And animals like these snakes, who could have a lifespan of 15 to 20 years, well, about 75% of them die within a year in captivity. And just a word of reassurance, as far as our beloved pets are concerned, there's far more risk of humans transmitting coronavirus to pets than the other way around. There is no scientific evidence at the moment to show that we can get sick from our pets as far as coronavirus is concerned. Yes, people who have coronavirus and who've got dogs or cats, you may find a little bit of, you may find some antibodies in your pets, but that does not mean that they are going to infect other people. So there, I think it's important to reassure people that, you know, our pets are quite safe. Animal research is a different story, and it is World Day for Laboratory Animals. So let's not forget the animals used in medical research in general, and in relation to the search for a vaccine or treatments for the coronavirus. It's highly unfortunate that scientists are using animals to try and mimic human disease. We've got so much information now, so much human data as a result of this pandemic, as a result of animal researchers being caught out and not having a so-called animal model ready. So what they've done is, so what a lot of clinicians have done is they've fast-tracked medications that were already known and tried and trusted in humans and have been on the market for a long time. So we know what their side effects are. We know that they have antiviral properties. So they've been used in combination with varying success. And as far as the search for a vaccine is concerned, well, there are already human clinical trials underway without animal testing, based on in vitro methods or other methods, human-based research methods that have shown that this vaccine can go to clinical trials. It's again very unfortunate, very sad that animal researchers are now rushing to find an animal model to give them the sort of the response they want. But I think this is a crucial time, and I think we can learn a lot from the coronavirus pandemic in terms of letting go of the animal model, in terms of stopping animal testing, because we can see that when animal researchers do not have an animal model, we can still make medical progress, we can still make scientific progress. And I think that's very important. So I think the way to go in this situation is prevention. Obviously, test kits to find out, for people who want to know whether they've already had the virus or not and are not showing any symptoms. We have a lot of human data. Let's use that. We have some amazing human-based test methods, in vitro test methods, like body on a chip, organs on a chip. We definitely need to stop using animal cells and genetically modified animal viruses to manufacture human vaccines. There have been some horrible accidents in the past because of this. Just one example, the early polio vaccine, which was developed around 1955, was discovered to contain SV40 virus. Now, SV40 virus stands for simian virus. SV is simian virus, in other words, a monkey virus that was naturally found in the monkey kidneys that were used to manufacture the early human polio vaccine. And this vaccine, or the virus, was not detected until about 1963. So between 1955 and 1963, millions of people received polio vaccine contaminated with the SV40 virus. Now, the SV40 virus is not just another virus. It's actually the most potent cancer-causing virus known to science. And so perhaps it shouldn't be a surprise that there's been an increase in the number of some rare human cancers where actually bits of the DNA of this virus were found. So very, very dangerous. Another episode in the late 1990s in the United Kingdom, polio vaccine again was manufactured using fetal calf serum. Fetal calf serum obtained from bovine, from cattle in the UK, just straight after the mad cow disease crisis. So there was a huge public outcry, and of course the government straight away took the vaccines off the shelves, and told people, don't worry, there's the risk is really infinitesimal. But if we can use human-based methods, if we can use cleaner cell techniques, which we can today, then surely we shouldn't be using animal cells to grow viruses for humans. So we should stop using viruses, genetically modified viruses, and animal cells to manufacture human vaccines. So this is an example. It's called the MIMIC system, which is an in vitro test method for testing and developing vaccines. It's very, very highly technologically advanced. And this is just one example. So it's one example to show how it can be done. So this is based on this MIMIC system, that not only will this more accurately model human diseases, it will also eliminate any ethical, moral, and scientific issues involved with the use of live humans and animals. An in vitro model turned MIMIC, which is a modular immune in vitro construct, was designed and developed to reflect the human immune system in a well-based format. The MIMIC system is a laboratory-based methodology that replicates the human immune system response. And then you have a link to an article, but if you just type in MIMIC in PubMed, you'll get several articles on that. So there we go. So thank you so much for joining us today. Thank you so much for listening to us. And now, if there are any questions, I'm going to ask Rachel perhaps to read them out. But first of all, I think I'm going to ask Rachel or Adam to just talk about this last slide.
Sure, thank you, Andre. So with all that information, what can you do in your life to help end animal experimentation? Well, you can search the Humane Charities list, which is supported by Humane Research Australia, and that helps you find charities who do not invest or participate in animal research, so you can be sure that your contribution is not inadvertently supporting animal use in that research. And directly with Humane Research Australia, there are a number of contributions you can make. HRA has recently joined a coalition of groups to thank and support research institutions around the world who have embraced non-animal methods. And you can add your name to the messages of support at humaneresearch.org.au. You can also find a bunch of case studies and campaigns to which you can send an email to research facilities and universities encouraging an end to their animal use. And of course, you can contact Humane Research Australia directly to volunteer or donate and become involved in actively promoting alternatives to animal use in research.
Okay, Rachel, if there are, I think, a couple of questions, if you like to read them out, and the ones that you'd like to answer, please go ahead. I'm not sure if Rachel is with us. Adam?
Oh, sorry. Hello.
There you go.
There we go.
Yes, sorry, I can see I've unmuted myself. We had a couple of questions in advance, and I can also see there's a couple in the chat and in the Q&A section. So I might start with the ones we've had in advance, and then we can move on to the new ones. So someone has asked, I'll read out the question. Do we have any best guess understanding of how many labs, unis or hospitals are currently using non-human animals or intend to use for COVID-19 issues, either vaccine or testing the disease implications and immunity, etc., including global number of animals and species type? So with responding to that question, I don't have an exact global number. My estimation would be at the minimum the tens of thousands, and the countries that we're most hearing about research taking place is the US, China, Canada, the UK, the EU, and South Korea. In terms of the animals that are being used, those that are being used to try and replicate humans are largely non-human primates, transgenic mice, ferrets, hamsters, and there's also animals that are being used to try and see whether other species can be impacted. So there is research that is being done that has involved dogs, cats, and farm animals. So I can't give an exact figure, but there is a lot of non-essential research that is being put on hold while this COVID-19 research is escalating, and there's continuously new calls for funding and new research taking place. So I imagine it's going to be very high. But that's not to say there isn't research taking place that's not using animals as well.
Any other questions, Rachel, that you want to answer?
I think there was a question asking about the situation in Australia and whether there's using animals here. And we know for sure that ferrets are being used at the CSIRO facility in Geelong. That's been quite widely publicised in the media. And there is a proposal to import more ferrets for that research. We have put forward a submission to try and stop that additional import of ferrets. You've probably seen on the news the institutions that are involved in the research. The ones that I'm aware of is the University of Queensland, the Peter Doherty Institute, Griffith University, Eliza Hall, and not all of their research is using animals, but the problem is that they're often working in partnership either with other Australian organisations or institutions overseas. So at some point, it's very likely that there will be research involving animals. So whilst their particular element may be trial of repurposing drugs in healthcare workers or human-based research at a later stage, it's likely that animals will be used. And I know it's ferrets in Victoria and in Queensland mice. But I did hear some positive news today about Griffith University, also in Queensland. They are using live human respiratory cells to test the drugs, and a quote from a spokesperson from that university is that when we're testing on them, that's the respiratory cells, we can already say it's more likely to get a similar result in a human. Of course, in people, it may be different again, but we think we can get there without having to go into animal models. We are far, far closer. So I think that's quite positive development.
Okay. Shall I take a couple of questions that have been posted at the moment?
Yes, please.
Okay, so somebody's said the term wet markets is also used in the live export industry as distinct from chilled or boxed meat. Could these wet markets also potentially carry zoonotic pathogens or viruses to humans? Yes, I mean, if we're talking about sheep or cattle being transported by ship, you know, spending several weeks at sea, well, some of the animals just die from stress. A lot of them, of course, pass diseases from one to the other. because they are in such cramped and stressed, unnatural conditions. So they're certainly passing diseases from one to the other. I'm not sure whether they are going to transmit new diseases to humans, but certainly their immune system is highly compromised. And I think, well, I think that by the time they reach land and then they are transported and then killed at the abattoirs, there is not as much contact between animals and humans as there is in the situation of a wet market, where there's lots and lots of animals on display and people, lots and lots of people mingling. So I think that's the answer to that one. Then another person says, not a question but a comment. I believe that there are ferrets being used currently for COVID-19 studies. That's true. Sadly, that's true. The ferret is very commonly used for testing and developing vaccines that are connected to the respiratory system. Here's one for you, Rachel and Adam. One of the first slides had the number of animals used in each state, but nothing from ACT. Firstly, how do you get these numbers? And considering the ANU and its rather large medical research facilities is in the ACT, is there a good chance quite a few animals are sadly used in the ACT?
I can attempt to answer that one. So we gather the statistics. Some states and territories provide the information and some don't. So for the ones that don't, we have to provide estimates based on the last time that they did provide that information. So it's always fairly conservative. And there is no national system of reporting. It's up to the discretion of each state and territory. And that's something that we're calling for, a national reporting system. But unfortunately, that's not in existence at the moment, and there's no obligation for them to provide that. Whether they collate it themselves, I'm not sure, and they just don't publish it. But each legislation is slightly different in each state and territory. So that's why we don't have the information precisely for every state and territory, only the ones that have published it.
Okay, then one of the listeners has asked, do you collaborate with PCRM, which is Physicians Committee for Responsible Medicine, and their campaigns against animal experimentation? I certainly do, and I know lots of organizations do. And of course, some of the information that we obtain from them is passed to HRA. I don't know if Adam or Rachel, you want to add anything to that?
Yeah, we do. They have a very good network for young researchers who are interested in alternatives. So when we have people who contact us and are interested, then I always put them in touch because it's a really good network. They have a lot of webinars and resources and training and scholarships and so on. So we definitely share information with them.
And here's another question for you, Rachel or Adam. Does HRA work with MRC at all to try and encourage them to change their grant funding decisions to find research that uses alternative methods of testing?
Yes, we are always in contact with them, and if we are profiling case studies that we feel should not have taken place, we always contact NHMRC. And with the COVID research, there has been some grant opportunities which are open for international researchers, and if that happens, I pass it on to the NHMRC for them to pass on to their networks. We don't have 100% response rate from them, but we try our best, really, and there are individuals who are probably more supportive within that agency than others, but we'll keep pursuing it.
Okay, then we have a question. When you say that animals should not mix, how safe are the animals in all the wonderful sanctuaries around the world that save so many unwanted animals? Well, I think that first of all, if the conditions are hygienic, if the animals are well fed, and if there is sufficient space for the animals to, go and lie down in a corner by themselves if they want to. In other words, the conditions are not cramped. And as I say, the animals are healthy and not stressed. I think the risk is small. I think there is always the possibility of species that do not naturally come into contact with each other. If they are forced into contact, and if they are kept in cramped and unhygienic conditions, I think that can lead to trouble. But I think for for a sanctuary where animals are loved and well fed and kept in hygienic conditions and have room to move around and not cramped, I think that's pretty okay. A comment: I'm currently doing my PhD on the consumers of pangolins in Vietnam. It is so frustrating to see wet markets open again, not only because humans clearly don't learn, but those poor animals don't deserve what we do to them. Humans have a lot to answer for, absolutely. And then I'm going to go to the conversation box. So we're getting questions both in the conversation box and the Q&A. So how safe is the flu vaccination? That's a question to ask your doctor. Why aren't statistics available for SA? Is that
I referred to that point earlier
okay.
in that all states and territories are different, but it's always, if you live in SA, it's always worth writing to the department and saying that you would like those figures to be available to put the pressure on them.
Okay. Then somebody asks, from what I've seen in the media, it seems that the Doherty Institute in Melbourne is working solely on human samples from patients.
Yep, I think they were working on the samples obtained from the first travellers returning back to Australia who were diagnosed. But my only concern is just that they are working in collaboration with other institutes that are testing on animals. So, but I think that particular research that they're doing is human-based.
Okay. What are the implications with such viruses in terms of antibiotic resistance? Okay, most antibiotics are aimed at bacteria, not viruses, but there are some antibiotics, for example, tetracyclines, which do impact on viruses. Yes, I think the overuse of antibiotics in the dairy industry and in the meat industry, all that could have a negative impact on our health in terms of viruses perhaps developing resistance to some of these antibiotics. And then we have another question: where are we in terms of stopping cosmetic testing on animals? I'm sure Rachel or Adam can comment on the situation in Australia. I just want to make a comment that it's the cosmetics testing issue is one of the success stories of the animal rights movement in the sense that consumer pressure and public opinion forced the cosmetics industry to put their hand in their pocket and develop a number of non-animal testing methods. Now, just for your information, now this applies to pharmaceutical industry and the cosmetics industry. There are around 11 tests that deal with toxicity. And what the cosmetics industry did is that they funded about 3 or 4 out of those 11 tests, which all have to do with cosmetics. In other words, skin toxicity. So how cosmetics can affect you by going through your skin. So that's what they funded. Unfortunately, they missed a golden opportunity to collaborate with the pharmaceutical industry to replace the other 7 tests, because those are actually some of the most cruel tests. So we're talking about acute toxicity. We're talking about repeat dose toxicity, etc., etc. So the cosmetics industry needs to replace animal testing in those areas. It hasn't done so yet. Rachel, I don't know if you want to add a comment.
Well, in terms of cosmetic testing on animals in Australia, there will be new legislation coming into place on the 1st of July this year. So it isn't technically a ban, it's really trying to restrict testing on animals. So it's preventing companies from using animal test data, but there are exemptions if the product is a dual purpose, so as well as cosmetic, has another industrial use, or so it's not perfect, but it's some progress, and unfortunately it's not going to apply to products which are imported from overseas, so it definitely has its limitations.
Okay, we have another comment here. The NHMRC has an animal welfare committee and is responsible for the review and preparation of the code of practice for scientific research and teaching involving animals. HRA, citizens and others can send feedback during the review process. Legislation was passed last year in July or August in Australia to ban animal testing for cosmetics. In reality, animal tests for cosmetics weren't common before the ban, but could be done on review at a state level, by a state level minister. I don't know if you want to add anything, Rachel?
No, that's all true, but it's not technically a ban on animal testing for cosmetics. It means that companies can't use the data from animal tests for new products after that date. So it doesn't mean that all the ingredients will not have been tested on animals at some point in the past.
Thank you. Okay, well, from my end, I don't see any more. Another comment has just come up. If we develop a vaccine for COVID-19, could a similar version of the vaccine be used to protect the mountain gorillas, as they are likely susceptible to complications arising from the virus as well? Would the vaccine need much adjustment to be used on gorillas? That's an interesting question. I think from now on, perhaps when people go to visit mountain gorillas, they must wear face masks and keep a good distance away from the animals. I think it would be tricky to to do clinical trials on mountain gorillas to find a vaccine. So I think, yes, I think we must just keep our distance from these lovely animals. We can't develop a vaccine for every single potential zoonosis out there. I mean, mountain gorillas are susceptible to some of common human diseases. So I think that the most important element here is prevention. So staying away from them, keeping our distance as much as possible. Yeah. I think we've dealt with all the questions so far. Do you want to add anything in general, Adam or Rachel?
I might just add that we're quite frequently posting articles on our social media about COVID research, both animal and non-animal research that's taking place. So if you follow that, you can keep up to date. We also have a podcast specifically on COVID-19 research, and it analyzes all the research that's been taken place and discusses a lot more in detail just further from this webinar. So you can listen to that as well. And any other questions, you can feel free to email HRA at any point or give us a call as well.
Okay, well, I see no more. One more message. Okay, so in addition, well, we have a thank you for an amazing webinar. Well, thank you for taking part and doing your part and making other people aware. Very, very important. I just missed a comment in an earlier question. The EU banned animal testing for cosmetics in March 2013. So I hope I made it clear that it's not a total ban in the sense that the 11 required toxicity tests have not all been replaced by non-animal testing methods. Only the 3 or 4 that are really that really concern the cosmetics industry have been replaced by non-animal testing methods, and it's up to us now to put pressure on the cosmetics industry and the pharmaceutical industry to validate, I'm using the word validate, non-animal test methods, because there are quite a lot of very, very efficient and reliable non-animal testing methods already available to replace those 7 nasty, horrible, cruel toxicity tests on animals. But as long as they are not validated, they cannot be used to replace, no government will touch them. So if we want to get rid of the animals used in regulatory testing, regulatory toxicology, those test methods need to be validated. Now, the industry, the pharmaceutical industry, the cosmetics industry is in no hurry to do so. They've had 30 years in which to find alternatives. And all they did was find 4 alternatives to the skin toxicity. So they're really not pulling their weight. They certainly have the funding to do so. In fact, they are far better placed to fund the validation, because the validation process can take a few years, and it's quite expensive. But the pharmaceutical industry and the cosmetics industry, they certainly have the money to do it. What is lacking is the will. And although we could use consumer pressure and consumer boycott to put pressure on the cosmetics industry, and it worked, we can't do the same for pharmaceutical drugs, because all pharmaceutical drugs must by law be tested on animals. So there's no choice between animal tested and non-animal tested pharmaceutical drugs. So that's where it's more difficult. But I think it's important, and I know that HRA has plans to campaign for the replacement of the remaining toxicity tests. Okay, well, I think we've handled all the questions. Do you want to sum up with anything, Rachel or Adam?
No, just thank you very much for everyone attending and for your support and for listening to us on a Friday evening.
Fantastic. Okay, well, very good evening to everybody out there. And as I say, we're going to, we should have this on YouTube fairly shortly if there's no technical problems. So best wishes to all, and World Day for Laboratory Animals 2020.